Well, Pluristem is a public company. It's an American company, although all of the employees of Pluristem are in Haifa, Israel, other than myself. And what we do that's unique is take the adult stem cells, the non-embryonic, non-controversial cells from the placenta, and which is the afterbirth, and then we grow them or expand them in a very unique proprietary environment. It's a three-dimensional environment, unlike any other university or company that is dealing in stem cells. So we basically have the best of all worlds. We're taking non-controversial stem cells from an organ that has traditionally been thrown away and are expanding them in a what we believe is the correct, unique way of doing that. We have several animal models of different disease states that we have tested these cells in, and I should back up and say that because of where we get our cells and how we grow our cells, we're able to take the cells and use them off the shelf, one size fits all, no matching needed. Our first indication, again, we have several animal models that suggest that our cells can be effective for a lot of disease states, and as a lot of the participants here at this conference are going to tell you, that the cells are anti-inflammatory. Our cells do not act by differentiating into other tissues. They immune modulate. They basically are involved in a cascade of cytokine and protein release that basically act as anti-inflammatory agents. And our first indication is in limb ischemia. It's where we have peripheral vascular disease, very common actually, patients that are diabetic, obesity, smokers. They basically have cholesterol plaquing of the arteries, primarily going down their legs. And our cells have been approved by the FDA and also the FDA equivalent in Europe to begin phase 1 safety trials on both continents, here in the US and in Europe. We've initiated those trials, and probably the primary reason that we were invited to attend this conference is to basically report about that clinical trial. It's a little different depending on which continent you're talking about, but essentially, using the same cells for limb ischemia. And the phase 1 trials, 2 locations in Europe, 2 here in the United States. One is going to be at Duke, and the other is in Alabama, the University of Alabama Associated Hospitals. The patients that will be entered into the phase 1 trial will be patients that have end-stage vascular disease. They either have rest pain because of a decreased blood flow to their muscles of their legs, or an ulcer that's not healing, or possibly gangrene of their toe. But bottom line is they're actually headed to amputation. They're headed to the operating room. So these are the patients that are going to be getting the cells in the phase 1 trial. The patients won't have to undergo any kind of procedure prior to getting the cells. Everyone will get cells. It's a uncontrolled dose-escalating trial. Between the 2 continents, there'll be 5 doses that will be tested. The patients will receive cells in the muscle of their affected limb, either above or below the knee, depending on where their problem lies. And the procedure has taken in about 20 minutes. The FDA has agreed with us that if we're going to have a problem with safety, it'll probably be a anaphylactic reaction, kind of like a penicillin reaction. That's never been reported in anyone else's stem cells. So we only have to hold the patient about 6 hours after injection, and then they can go home. It's all outpatient procedure. And then they come back periodically for assessment. Again, the primary objective is adverse events, but we are going to be collecting information concerning blood flow to their extremity, wound healing if they have a wound, quality of life issues, pain assessment, those kinds of parameters we're going to be collecting, that information we're going to be collecting as well. Between the two continents, there are going to be about 30 patients that are going to be treated. And internally, we believe that we should be able to have the study completed and the information assimilated in about a year.